All right, I'm going to do a quick run through of a skeletal muscle contraction. Remember, this is a muscle cell in skeletal muscle. So we're going to start with the neuron. The neuron is receiving a signal from the brain. So we have an efferent nerve, and the signal is sent with sodium ions. And when the sodium ions travel all the way down, when it gets to positive 40 at the axon terminal, that's going to open the calcium gate. The calcium is going to come in, and remember, calcium moves things. So the calcium binds to the vesicle containing the neurotransmitter. The neurotransmitter is acetylcholine, and an enzyme called CHAT sticks the acetyl to the choline to make acetylcholine. The calcium binds to the vesicle containing acetylcholine. It's going to move it to the cell membrane of the neuron, where a protein called SNARE is going to help the acetylcholine cross the cell membrane into the synapse. Out in the synapse, it is going to attach to the acetylcholine receptor, which is a ligand-gated ion channel. That means when the ligand, acetylcholine, binds to it, it opens an ion channel for an ion, which is a charged molecule, in this case, sodium, which is you're going to see the same story as you saw before. So the sodium is going to come into this T-tubule, which is very similar to the axon of a neuron, but we're in a muscle cell now. The sodium is going to come in, it's going to get to positive 40, and that's going to open a calcium gate. Same story as before. And calcium moves things. This calcium gate is on the sarcoplasmic reticulum, which is full of calcium. The calcium comes out into the cytoplasm, and it's going to help move our filaments here, our muscle filaments. We have the thick filament and the thin filament, and those two filaments are going to slide along each other to contract the muscle. It's called sliding filament theory. The thick filament is myosin, the thin filament is actin, but it's not that easy because we can't have myosin binding to actin all the time. So we have it when the cell is at rest. We have actin is covered up by another protein called tropomyosin, which is pinned in place by troponin. So troponin is like a receptor, and it's very, very negative, right? So we're going to put two negatives there. And because I want you to see that calcium is going to be attracted to bind to it. Calcium is, of course, 2 positive, so +2. But two other ions can also bind to it, potassium and magnesium. And I want you to notice potassium has 1 charge and magnesium has 2 positive charges. So magnesium is going to bond a lot stronger to troponin than potassium. And when potassium or magnesium are bound to troponin, calcium can't bind, so the muscle can't contract as much. And so potassium and magnesium are considered muscle relaxers. And magnesium is going to be a much stronger muscle relaxer than potassium. Also because there are going to be potassium leak channels that where the potassium leaves. We're going to learn more about those as we move through. So what happens is the order is the calcium binds to troponin and moves the troponin, which moves the tropomyosin, exposing the actin. The myosin heads bind to the actin and pull it together. All right, so I'm going to say that again. The calcium binds to the troponin, which moves the tropomyosin so that the myosin heads can bind to the actin and pull the filaments together. And that requires ATP, that is contraction. The whole muscle gets shorter and thicker. In order for it to relax, the muscle to relax, the calcium pump on the sarcoplasmic reticulum uses a whole lot of ATP to pump that calcium where it doesn't want to go back into the sarcoplasmic reticulum. And when the calcium leaves the troponin, the actin and myosin are going to slide back to their starting positions in a relaxed muscle state. Also, don't forget, acetylcholinesterase is an enzyme that's going to come through the synapse and is going to break apart all the acetyl and choline so that the acetylcholine doesn't continue to stimulate the muscle cell. And the acetyl is going to get recycled, so is the choline. And the choline goes through a pump called the choline or cholinergic reuptake pump on the neuron to go back and be made into more acetylcholine.